Dear viewers, greetings. In this video, we are going to see about Lyophilizer. The contents of this video includes an introduction about Lyophilizer, principle of Lyophilizer, types of Lyophilizer, instrumentation of Lyophilizer, steps for using a Lyophilizer, applications of Lyophilizer, advantages of Lyophilizer, limitations of Lyophilizer, and precautions for using a Lyophilizer. Lyophilizer. A lyophilizer is also known as freeze dryer. It is a device used to remove water or other solvents from a product by the process of sublimation. A lyophilizer executes a water removal process typically used to preserve perishable material, to extend shelf life, or make the material more convenient for transport. Lyophilizer work by freezing the material, then reducing the pressure and adding heat to allow the frozen water in the material to sublimate. Lyophilization technique is widely used for preserving perishable material, improving the shelf life of pharmaceuticals, and preparing biological samples for research. Principle of lyophilizer. The process of lyophilization involves a phenomenon known as sublimation. An object that is liquid passes directly from one state to another, unlike an object that is solid, for example, ice. Lyophilizer involves removing the water from the frozen state material and then heating it by conduction or radiation or even both to the point that the evaporated liquid leaves only the solids or dry components of the original liquid. Lyophilizer is necessary to dry materials at low temperatures and pressures than the triple point. Types of lyophilizer. There are three types of lyophilizers. They are batch freeze dryers, manifold freeze dryers, and rotary freeze dryers. Types of lyophilizers. The first type of lyophilizer is batch freeze dryers. The batch freeze dryers are used for handling a large batch of products at once. These batch freeze dryers are ideal for pharmaceutical products like vaccines or bulk materials. The second type of lyophilizer is manifold freeze dryers. The manifold freeze dryers are typically used for small sample sizes or individual vials. The samples are frozen individually before drying while using this manifold freeze dryers. The third type of lyophilizer is rotary freeze dryers. The rotary freeze dryers are used for more delicate samples that require gentle drying. The rotation prevents uneven drying and structural damage to the samples. Instrumentation of lyophilizer. An instrumentation of lyophilizer contains 6 major parts. They are chamber, condenser, vacuum pump, cooling system, heating shelves, and control system. Chamber. Chamber is the main compartment where the product is placed for freezing and drying. Condenser. The condenser traps the sublimated vapor and converts it into solid ice, preventing it from returning to the product. Vacuum pump. The vacuum pump reduces the pressure in the chamber to facilitate the sublimation process. Cooling system. The cooling system lowers the temperature in the chamber to freeze the product. Heating shelves. The heating shelves gradually warms the product during the primary and secondary drying stages. Finally, control system. The control system allows operators to monitor and adjust temperature, pressure, and drying time. Steps for using a lyophilizer. There are 6 steps. The first step is preparation of the product. The material to be freeze dried is placed in containers such as vials, flasks, or trays. The second step is freezing. The product is cooled to temperatures below its freezing point, usually between -50 degrees Celsius and -80 degrees Celsius. This ensures the water in the product is converted to solid ice. The third step is vacuum application. Once the product is frozen, a vacuum is applied to lower the pressure within the chamber. The fourth step is primary drying or sublimation. Heat is gradually introduced to sublimate the ice and converts the ice into the vapor. The condenser traps the vapor, turning it into ice to prevent it from returning to the product. The fifth step is secondary drying or desorption. The temperature is slightly increased further to remove bound water molecules from the product and leaving it with minimal moisture content, often less than 1%. The sixth step is sealing and storage. The dried product is often vacuum sealed or stored in airtight containers to maintain its stability and shelf life. Applications of Lyophilizer. In pharmaceuticals, lyophilization is commonly used in the production of vaccines, antibiotics, proteins, and enzymes to improve its shelf life and ensure stability during transportation. In food industry, freeze drying or lyophilization is used to preserve fruits, vegetables, meats, and coffee, allowing them to be stored for extended periods without refrigeration. In biological samples, in research laboratories, biological samples like tissues, bacteria, and enzymes are freeze-dried for long-term storage and later rehydration without loss of activity. In cosmetics, lyophilized ingredients are used in high-end skincare products to preserve their effectiveness and ensure long shelf life. Preservation of living organisms. Certain microorganisms like probiotics are freeze-dried to maintain their viability for the use in health supplements and therapeutic products. Chemical and pharmaceutical stability. Freeze drying is used to stabilize sensitive chemicals, biologicals, and pharmaceuticals that degrade in the presence of water or during traditional drying methods. Conservation of historic artifacts. In conservation science, lyophilization is sometimes used to preserve delicate artifacts, manuscripts, or archaeological specimens by carefully removing moisture without causing structural damage. Advantages of lyophilizer. The advantages of lyophilizer includes extended shelf life, preservation of structure and function, lightweight and easy to transport, minimal heat damage, rapid rehydration, preserves chemical stability, and ideal for biological and pharmaceutical applications. Limitations of lyophilizer. The limitations of lyophilizers includes high cost, time consuming, not suitable for all materials, energy intensive, specialized storage required, and potential for structural changes. Precautions for using lyophilizer. Pre-freeze the product. Ensure the product is completely frozen before starting the drying process to prevent sublimation from occurring unevenly. Avoid overloading. Do not overcrowd the chamber as this can obstruct proper airflow and lead to inefficient drying. Monitor temperature and pressure. Keep a close watch on temperature and vacuum pressure to ensure optimal condition for sublimation and prevent damage to the product. Proper sealing. Ensure that the vacuum chamber is properly sealed to maintain the low pressure environment necessary for freeze drying. Use compatible materials. Only use containers and trays that are suitable for freeze drying, as some materials may crack or degrade under low temperature and vacuum. Avoid overheating. Gradually apply heat during the drying phase to prevent rapid temperature changes that could damage sensitive products or alter their properties. Regular maintenance. Perform routine maintenance on the lyophilizer, including checking for leaks and replacing filters to maintain equipment efficiency and ensure product quality. Finally, handle hygroscopic materials with care. Products that easily absorb moisture should be handled carefully post-drying to avoid rehydration during handling or storage. Dear viewers, that's all about the Lyophilizer or freeze dryer. Thank you for your support. Thank you.